Hey, actually, in the last classes, we have discussed about the structural chromosomal aberrations, and I will discuss numerical chromosomal aberrations by taking a wheat as a case study. Because we know that wheat is a typical, most of the cytological studies which happened in the wheat, and we have like some classical examples of using these techniques, cytological techniques, and using these numerical aberrations for resistance breeding in a wheat. It's actually a classical case study. That's why I'm discussing wheat. Uh, as well, a parallel, I will cover the wheat crop breeding and also the numerical chromosomal aberrations. So before that, we will have a short introduction about the wheat. Wheat is a major staple crop of the world, and that to India is also in northern part of India is a wheat is a major staple crop. In India, is actually earlier you can see that the area you can see thus 25.38% of the area was increased, and the world it's actually decreased by 2.73 million hectare. Actually, this is the old data. Okay, I was prepared the slides last year. You just see the estimates of 2024, how much production was increased. Actually, even the world average is decreasing, but Indian production is increasing. That's what I want to say. But then, when you ask, like, take the production, like when you compare with the Green Revolution area, we have increased 73% of the increase we got from the 1950s to now 2024. This much area of production was increased, per area production increased because of Green Revolution that we all know. And the productivity is increased 2483 kg to 3498 kg per hectare area, 40.9% increase during the time from the Green Revolution. The now world increased from 862.9 kg per hectare area. It means to say that Indian wheat production is very good in a better condition than the world average. And please, this is estimates of 2023, like up to, but we just see the estimates of 2024. Before going to that, like we have many questions from the wheat and meat actually, and wheat is most of the questions are asked in the exams, like from series 2020 to 24. Most of the questions are from the wheat. At least you can expect of 2 to 3 questions will be there in every exam, because wheat is our staple crop of the northern India. Okay? An Indian wheat variety CV591 was developed by, it was 2023, I think, was developed by Ram Dhan Singh. Then, Triticum aestivum is actually a common bread wheat, is hexaploid in nature. That's why I have taken a case study, taken as a case study first to study the numerical chromosomal aberration. See that it is a Then the genomic constitution of the bread wheat. See, the questions are repeating. See, the Triticum aestivum is hexaploid and it is AABBDD. It is asked in 2022. Questions will be very simple. No need to worry about like uh, in detail about any topic. Then Pavan, Veri, Bovide, and Attilia and Coach, the lines which have been extensively the breeding program of each crop. Actually, if you don't know anything, we know that Veri series of wheat. It's very very famous. By looking at this one only, we can know that it's a wheat. Sorry, it's a wheat. Okay. And in rice, we have discussed so many lines were there. You can see, actually, these were the varieties which are across the world they are cultivating red wheat. There are several wheats were there, very important to remember, like Pavan wheat, Bovite wheat, Ataria wheat. From these wheat only nowadays all wheat varieties are developed actually. That's why these were asked in the exam. Then origin and evolutionary pattern of wheat. It's a typical example of the evolution of polyploids. How the polyploids are evolved, it's a typical example. Okay, wheat evolves in involves Wheat evolution involves mutation, polyploidy, there is amphipolydy, and intergenic hybridization. And we'll see one by one like how what is intergenic hybridization. Through intergenic hybridization, how today's wheat is developed. And three main groups based on the chromosome number. We have diploid wheats, we have tetraploid wheats, we have hexaploid wheats that we will discuss one by one. You look at the genetic composition, nowadays the cultivated wheat having three genomes A, B, T, and each genome is derived from different donors during the evolution. And another wheat variety that is hexaploid, like Triticum aestivum and Triticum uh, jacobacus. It is also asked many times in the exam. What is the genomic constitution of Triticum jacobacus? It's actually AAAA genome. Normally we confused with ABG. It's not ABG, it's AAG. G two like tetraploid of A and one addition of GG. You can see the questions like tetraploid wheat species, Triticum dicoccum, is shortering in nature. The shortering nature because of brittle racid gene in a homozygous recessive condition. This we will discuss after discussing like how wheat is evolved during the evolution. See, like AA genomes, Triticum monococcum and Triticum urartu, like actually still it is debated, but as of we'll see this. Triticum monococcum is the donor for AA genome. And actually, earlier its wild species, Triticum monococcum agiloptoides, it's slowly cultivated during the domestication. Then it will become Triticum monococcum species monococcum. It is actually the donor of their present day wheat for the A genome. Then again, there is still doubt in Agilop's peltoids, which actually contributor of the B genome. Still, it's in a doubt. But uh, many times it asked in the exam the B genome, there is still doubt because of through genome analysis. We are not getting a proper hybridization. Like we can't able to trace back to this genome, like Triticum peltides, like Agila peltides. That's why still they are believed that B genome contributor is Agila peltides, but still it is in the doubt. And again, Triticum toschi is one of the important source of the D genome, Triticum toschi. It's a very important source of D genome. Through Triticum monococcum, Agilis peptides, and Triticum toschi is combinedly they will forming ABD genome. Okay. Again, Triticum Agilis peptide and Triticum urartu actually given Triticum turgidum, which is Triticum turgidum dicoccum. Then again during cross with Triticum toschi, then there is development of ABD. There are several theories. Okay. For example, if you can take Triticum urartu combines with Agilex peptides with the Agilex peptides, then there is a formation of Timophoebe GGAA, Triticum timophoebe. Still is a source of male sterility in the wheat. Then again it combines with again Triticum monococcum. There is a formation of Triticum jukovaski. There are several theories that we will discuss in detail. You can see here the monococcum species we having Triticum monococcum L and Triticum urartu. This is a monococcum sect and urartu sect. And you should also know many times asked in the exam what is the common name of this one. You can see the Triticum monococcum. The common name is cultivated anicorn, and Triticum sinskajae, it's a naked anicorn, and Triticum boeticum is a wild anicorn. Triticum vararth is a wild form. Okay. Then the tetraploid species includes Triticum turgidum, where it is called as wild emmer wheat. 20, Triticum dicocoides is a wild emmer wheat. Triticum dicoccum is emmer wheat. It's asked in the exam. Please remember. Triticum wild emmer wheat is Triticum dicocoides, where emmer wheat is Triticum dicoccum. And Georgian wheat is Triticum cremastivaceae. And please, this is not important, but this is Triticum dicoccum is most of the in South India we are cultivating it. Then Triticum turgidum is also called as rivet wheat or cone wheat or pollard wheat. Please remember, many times it asked in the exam. Then Triticum durum, it is called as macaroni wheat, and Triticum carthilicum, it is called as Persian wheat. Triticum turnicam is called as Croatian wheat, and Triticum polonicum is Polish wheat, and Triticum ethiopiacum is Ethiopian wheat. Triticum timofilus is very, very important because it's the source of male sterility. It is also called as Janduri wheat. And Triticum Ararticum jacobus is a wild janduri. Please at least remember some of them: unicorn wheat, emmer wheat, durum wheat, okay, rivet wheat, and polish wheat, janduri wheat. These are very, very important. Then another, like we having bread wheat is known as Triticum aestivum, club wheat is Triticum compacticum, and Indian dwarf wheat is Triticum sparacoccum. It also asked many times in the exam. So, and what is the common name of Triticum sparacoccum? It's an Indian dwarf wheat. And Triticum petrovalsky, it's a rice head wheat. It is also asked in the exam in the ASR unit. And matcha wheat is also asked in many times, but you can easily remember. And dinkel wheat, it is asked in the several times in the exam. Okay, what is dinkel wheat? Actually, in our exams, like it is commonly asked question. What is the common name of triticum spelta? It's a dinkel wheat. Please remember, because if wheat breeder prepares the question paper, and then definitely he will ask the question. Then yeah, another, like there are Zhukovsky wheat, triticum Zhukovsky, and these are the very, very important. No, they don't need to remember sect, all those things. At least you remember the common name and scientific name and the classification: diploid, tetraploid, and hexaploid classification. Which wheat comes under hexaploid, which wheat comes under tetraploid, which wheat comes under diploid. So I have given the detailed table, and also I shared the book also. You can see that one. Triticale, we'll discuss Triticale later. Then we know that A genome from Triticum monococcum and Triticum urartu is still debated, and the BB genome is closely related to Cetopsis section of Agilopsis pletoides, but still it is also in the question. And the GG genome is from the Triticum timophoebe, and DD is from Triticum Agilopsis toschi. And early cultivated were diploid, originating in the southeastern Turkey. Please remember the origin. Then the domestication focused on non-brittle rachis and null hatches. See, this one asked directly in the exam. You can see this question. Okay, non-brittle nature of this one. Then during the evolution, during the domestication, they actually lost from. That's why wild dicoccum species having um, tetraplets, which is a criticum dicoccum shattering nature. No, during the domestication, the shattering nature was lost. Okay, that's why. But this one is correct. Now it is in recessive condition, but earlier it is a dominant condition. Now they are non-shattering in nature. Okay, then what I was then a genome sources to monocom to Murartu, and actually the floor properties of wheat that we are discussing actually it is contributed by the D genome. It is also very important questions because nowadays floor properties, stuffing quality, and all those things are very, very important in bread making quality. And these quality properties were actually given by the donated by the D genome. It's very, very important. And we have discussed like what are the common name of this one, and like tetraploid wheat resulted from natural crossing between Urartu and Agela spletitis. Then AABB genome and Triticum timophoebe is AAGG genome. Then how Triticum turgidum? Actually earlier it was a white. Then actually Triticum dicoccum, how it is selected from non-brittle spikes. See, in this question they were asked like Triticum dicoccum is a brittle. It's not true. Triticum dicoccum actually it's a non-brittle spike. Then, during the domestication, these genes were lost. Then there is a artificial hybridization. Actually, M.K. Fadden and Sears had tried this hexaploid wheat from Triticum dicoccum and Digella toschi that we will discuss critically in coming classes. What is triticale? Actually, when they have crossed Triticum durum in Sagilla stopchi, or Triticum durum in Sicale seriale, Triticum durum with Agila surartu, what will be their answer? It is actually the man-made cereal which actually crossed by the rye and the wheat. Triticum durum and Sicale seriale, we have developed Triticale. Okay. Then you can see the another question. Why this were very, very important to learn about this table? You can see this question. An hybrid was developed by crossing the bread wheat and the coffee wheat. And the meiosis of the F1 plant, there will be a formation of. You know that coffee wheat is a tetraploid wheat actually. Okay. Should know what is the tetraploid wheat. When tetraploid is crossed with hexaploid, what will be the genetic constitution? Okay. You're at 7 and 14. Okay. 7 bivalents and 7 univalents. Okay. That's why you should be able to answer in such a way that when we will see this in detail, then because it's a Carphyl is tetraploid. The only confusion is we don't know either Carphyl is a tetraploid or hexaploid. That's why we can't able to decide. If you know this is a tetraploid and this crossed with hexaploid, definitely this will give 7 univalent and this will give your 14 univalents. So that if it is cell with 7 bivalents will be formed and 14 divalents will be formed. Based on that, you should be able to answer the question. Then, to take domesticated MRV. See, this directly asked. See, domesticated MRV. Wild MRV is to become dicocoids. Your domesticated MRV is to become dicocum. It become turgidum dicocum. Okay? Dicocoids is wild. Durum is another different type. Polonicum is different weight. Then, the most commonly used sterile cytoplasm of hybrid wheat breeding program is transferred from, as I told, it is a AAGG genome. It is a Triticum timophoebe, and it is a source for Triticum zygovasque hexaploid wheat. Then, when you look at the wheat inflorescence structure, it is commonly known as ear. The inflorescence is known as ear. It's botanically terminated terminal distichous compound spike. Actually, zigzag rice is the very unique feature of your wheat when compared to rice. We're having three anthers only in the rice. Then, in tetraploid hexaploid with ratchet stuff and resist disraticulation, but Triticum dicocoids and Triticum dicocum, disraticulation is easy. Okay, then each spikelet subtended by two sterile and empty glumes arranged alternatively in opposite side of short central axis. This is called as rachila. We'll see the look at see the how the rachila will looks like. There is an alternative arrangement of the glumes and with a large spikes. This is called as spikelet and alternate arrangement of rachis. And we having only three anthers and the stigma is feathery, bifid feathery, and we have lodicules. Then the genetic diversity is crucial for successful breeding programs, and we had to maintain the gene banks. And we will see that how we can conserve this one, either in situ conservation or ex situ conservation. And the majorly the one institute is working very hard to conserve all the germplasm sources, the meat. Okay, and through where actually the green revolution started, where Norrington revolutionizing wheat has been introduced in the Mexico. Then how we can analyze genome analysis? We have in the syllabus genome analysis of each and every crop is very very important. Like in 1918, Carl Sachs determines the chromosome number of durum wheat, 2n is equal to 28, and in 1918, Sakamura identifies chromosome number of 8 wheat species. And these findings were confirmed by Kihara, is actually the very very important scientist who used this technique. We have discussed this technique to predict the genomic relationship between the Triticum aestivum with their progenitors. Then he crossed alloploid wheat with diploid ancestors to create triploid hybrids so that by analysis by using this technique, he predicted who is the ancestor of today's cultivated wheat. Then, when you look at the cytogenetic stocks in the wheat, as I told, wheat is a case study for the numerical chromosomal aberrations that we are going to discuss today. Wheat aneuploids developed by Sears. Please remember the Sears is a very, very landmark like important scientist made landmark discoveries in the field of cytogenetics in the rice. Sorry, in the wheat. Actually, here Sears using cytogenetic techniques in the Chinese spring cultivar. He majorly works in the Chinese spring cultivar. He created so many monosomic lines, so many disomic signs, so many nullisomic lines, so that he used for transferring of some resistant genes or some quality genes for the cultivable wheat. And why the cytogenetics is most successful, cytogenetic techniques are more successful mean because of the polyploidy nature. Even though you can create monosomics, it can able to tolerate, it can able to survive. But you create monosomics in diploid, what will happen? They will not going to survive. That's why cytogenetics is more successful in the wheat rather than any diploid cultivars. Haploid plants are identifying the cross between Chinese spring or rice. Then, what are the type of aneuploids lines for present? Before going to that, we will have a short discussion on what is the basis of numerical chromosomal aberrations. What are the types of numerical chromosomal aberrations are present in the organism? We will discuss first, and we will then go to the what are the things are present in the meat. Sorry, wheat. Actually, numerical chromosomal aberrations mainly classified into euploids and aneuploids. Euploids and aneuploids. Okay. Then, what is euploid and what is aneuploids? Okay, aneuploid means the loss of or gain of a chromosome number when compared to your diploid complement. Please remember this definition. Aneuploids. Many times that's the exam. When you look at the euploids, it's the entire set of chromosome either duplicated or it is lost. Okay? It is just regarding only one or chromosomes missing. But in the case of euploids, the entire set, the presence or complete genome presence or absence of complete genome from the diploid component of the set. Okay, there are again several classification we will discuss. I will make one slide and I will write the points. I couldn't able to make the typings. We will write one by one so that you will get the clarity. Okay, first we will discuss about what is the classification of under the aneuploids. Then we will discuss what are the different things for there. Then first you just observe in aneuploids, first numerical chromosomal variation. Sorry for the spelling. That's why not right. We have two types, euploids and aneuploids. And in the euploids, we having a lot of things. Tetraploids, triploids, haploids, diploid. Diploid is normal, and triploids, tetraploids, hexaploids. We have so on. In aneuploids, we have monosomic, nullisomic, tetrasomic, and so on. We'll discuss one by one. Okay. Then, what is aneuploidy? First, we'll discuss aneuploidy, then we'll go for. Actually, I'm starting a new way of teaching, like how say. Like aneuploids means loss. We have discussed loss or gain of a chromosome from the normal diploid component. First, we'll discuss monosomy. Is 2n minus 1 means that loss of one chromosome, loss of 2n minus 1, loss of one chromosome from the diploid set. Okay, There are typical examples, and this monosomics will not survive. It's rarely survive, will not survive in the diploids. Even though it survives, it's very, very difficult. But that's why monosomics are more common in the polyploids than the diploids. Then, what is the possible number of monosomics present? This typical questions were asked many times. Number of monosomics present in a species, number of monosomics possible in the species is equal to n. Okay? For example, you take wheat. How many number of monosomes are possible? We know that n is equal to 21, means 21 monosomics of wheat can be possible. Many times asked in the exam. Then this is about the more like what is the number of monosomics present in the wheat. And we have another classification among the monosomics. One is double monosomic. Is 2n minus 1 minus 1, and we have triple monosomic. Insert 2n minus 1 minus 1 minus 1. See the loss of chromosome from one and another set. Here the loss of chromosome from the three set means that there is a random between. For example, there is a one terminology called as univalent shift. What is this? What is the univalent shift? It means that when a univalent or unpaired chromosome interfere with the pairing of the other normal homologous chromosome, you can see here, if there is a 2n minus 1 for A chromosome, in the next time 2n minus 1 for the B chromosome after like a division or segregation. This shift in the monosomics from one chromosome to other chromosome, it is called as univalent shift. And this univalent shift is a typical character of your monosomics. Please remember this way. Then what is the application of this monosomics? It can help in the locating the genes, helps in the chromosome mapping. Writing is very difficult. Chromosome mapping. We will discuss how we can map the chromosome by using this one. Then physical mapping that we can do. This is about the monosomics. We have seen what is monosomic, types of monosomics, and number of monosomics can be possible. It's n number of monosomes can be possible, and what is univalent shift, and what is the use of monosomics. Then we can see that in wheat we have a lot of monosomics. Monosomics 21 total. The lines having single chromosome missing 20 plus one missing chromosomes, and monosomic with isochromosome. These lines having single chromosome missing, isochrome present, one plus missing chromosome and one isochromosome. In trisomic, we have trisomics also in wheat. The lines having extra copy 2n plus 1. Nullisomics also we have 2n minus 2 we have tetrasomics also 2n plus 4 or minus 4. This many times asked in the exam. We will discuss several questions rather than discussing the repetitive things. You can see how many number of nullisomics are possible with a plant with a chromosome number 2n equal to 20. How much? It's a n. Because we see that number of monosomics possible, is equal to n. So that here 2n is equal to 20 divided by 2, n is equal to 10. Very simple questions they will ask. This asked in 2022. Then you can see what is the total number of nullisomic lines possible in Nicotiana tabacum. See, this is also very, very important because we know that it is also n. Number of nullisomics also formula n. We have Nicotiana tabacum, it is 48. means 24 chromosomes will be present. See, this is asked in 2023. See, repeatedly they will saying questions were asking. This year also in 2024 also, they will just ask number of nullisomics only, number of monosomics only. Okay. Apart from this, they will not ask any in detail questions. If you remember this much, it's more than enough. Then we have nullisomics and we have discussed monosomics, and we will discuss what is nullisomy. Nullisomy means it is just 2n per 2 minus 2. Entire diploid chromosome is lost from the your 2n condition. That called as nullisomies. It is not. It is also not able to survive in the diploids, and it can only survive in the hexaploid conditions. Means possible number of nullisomics but in any organism is n. Okay, haploid chromosome number. Okay, another very important gene is called as ph gene, which actually leads to that we are going to discuss. Okay, I will discuss later. Then this is about nullisomy. And what is trisomy? Trisomy is 2n plus 1 condition. If there is a presence of an extra chromosome along with your diploids. And this trisomy is a very, and what is the source of trisomy? Triploids are very important source of trisomics. We have different type of trisomy. Primary trisomic, we have secondary trisomics, secondary trisomic, we have tertiary trisomic. What is primary trisomic? Because the additional chromosome is the normal chromosome. And what is the possible number of primary trisomics? It is n only. Okay, if it is 21, in week 21 trisomic, primary trisomics are possible. If it is a secondary trisomic, it is number of trisomics possible is 2n. Okay, because in secondary trisomics, the additional chromosome is not normal, it is isochromosome. And number of possible secondary trisomics are 2n only. Then the tertiary chromosome means here the additional chromosome is any translocated chromosomes. Okay, then the possible number of tertiary trisomics are infinity. Please remember the number of primary possible number of primary trisomics is n, number of possibly secondary trisomics is 2n, and number of tertiary trisomics is infinity. If you know this much is more than enough. And there is again further classification double trisomic 2n plus 1 plus 1. Okay, please remember this condition double trisomic, primary trisomic, secondary trisomic, and tertiary trisomic. What is the possible number of trisomics are possible in each case, and what is trisomics? If you remember this much, it is more than enough for the exam. Another one is we have tetrasomics. I will not going in detail about like what is the breeding behavior, what is this happen. It's unnecessary. If you want to read this, read it from the medicine book. It's more than enough. Then, um, what is tetrasomy? Heterosomy is a condition of 2n plus 2, where one pair of extra chromosome is present. It is exactly contrast to the allosomy, where entire one pair of chromosome is lost. But in the case of tetrasomy, it's having entire new chromosome is present, new pair of chromosome extra added to the diploid conditions. Then it is also only sustainable in the polyploidy. And then the another important uses of these aneuploids, we have seen monosomics, nullisomics, trisomics, and tetrasomics. We will study what is the uses of these aneuploids. One is to study the role or loss of gain of chromosomes. For example, we create monosomic lines, can study the gene. It's also called as breeding at chromosome level. Breeding at chromosome level. Means, for example, you create monosomic lines for one or the other lines. For example, we can create, for example, how many number of monosomic set can be possible in with 21. We can create 21 stalks of monosomic lines for each chromosome. You can create, then you can phenotype it. Based on that, we can see on which chromosome. For example, if 2n minus 1 for the first chromosome, then you can study the phenotype. Okay, what are the changes happening in this phenotype because entire first chromosome is not present. Then you again another plant 2n minus 1 for the second chromosome. Then you study the plant because the entire second chromosome is missing from this diploid. Then you can study, okay, what is the changes in the phenotype. Based on that, we can create the monosomic line. We can breed at the chromosome level. This is the one of the typical example of monosomics. We can study several examples in the wheat so that we will get to know how monosomics can be used to used in the used in the patient breeding. You can see that the transfer of LR9 from the Agilaps ambulata to Triticum aesticum using Triticum dicotyledonus as a breed species. And it is asked many times in the exam, and it's a classical examples, many times asked for 10 marks in the ASRB mains. Okay, then you just observe Triticum dicotyledonus we're having AABB genome which having 2n is equal to 28, and it's crossed with Agilis ambulata which is a donor for your LR9 gene. Then it is the genome is UU. Whenever you are crossing, you are creating ABU line, it's a having completely sterile. Then you have to do chromosome doubling AABBU. Then it is crossed with Triticum aestivum. Here the Agilaps ambulata, or Triticum dicotyledonus is a bridge species. Because Agilaps ambulata, if you want to cross with Triticum aestivum, it will completely sterile. You can't make anything. That's why you use the bridge species. First, you cross Agilaps ambulata as a donor species to Triticum dicotyledonus. You create an intermediate line. Then you cross with Triticum aestivum. Then reiterated back cross with Triticum aestivum. It creates the monosomic align addition lines A, B, B, D, D, and U. Okay. Then this can be used for the further um, resistance breeding. This is a typical example of transfer how LR9 is gene transfer, and the variety is created is called as your transfer. The variety name itself is called as transfer. It asked in the 2022. Please remember this. Then see the example. It's an alien translocation line carrying the segment of a transfer is a variety which are developed by alien translocation of carrying the segment of Algilla symbolata. And the line was found to carry LR24. So, no, it is the line, the transfer variety which having LR9. Okay? Very, very important, these things. Then when it comes to monosomic, this one, we have double monosomics, we have diteleosomics, we have double diteleosomics, diteleo-monoteleosomics. What is this teleosomics? Okay, because these lines are missing in a short arm or long arm of the chromosomes, 20 normal chromosome plus one teleocentric chromosome. The additional chromosome is teleocentric chromosomes. The teleocentric chromosomes has only one arm. That's why we can create a single diteleosomic lines or double diteleosomic lines or diteleo-monoteleosomic lines also. There are many types of lines are developed. Then we can see the what is align addition lines and what is align substitution lines. Means that because of developed alien addition and substitution lines in wheat, it's normally taken place in 1950 by Sears. For example, you cross hexaploid wheat with a diploid alien species to create an amphidiploid, and cross an amphidiploid with an hexaploid wheat to get an heptaploid. Self-heptaploid potentially produce monosomic addition lines, and self-monosomic addition lines get disomic addition lines. Then it can be applied in wheat dry hybridization lines, and it can be used in the resistant breeding that we have seen here. How the alien addition line can be developed. You can cross with the tetraploid, then you can create the intermediate line. Again, you cross with the tetraploid. Again, you go for continuous crosking. See here we having the alien addition monosomic line. Okay. Then what is diploidization? Very important mechanisms. Wheat, even though it's a polyploid, but it does not undergo any multivalent formation. And why it is so? Even though it's a polyploid, why it is not go multiploidation? Because the even the polyploid chromosomes will act like diploid. This is called as amphidiploid. And this diploidization mainly due to there is a absence of homologous pairing between the homologous pairing between the different genomes A, B, D genomes. There is no pairing between A and B and D genome. This is because of the presence of PH gene in the B chromosome. Okay, this PH gene is very important in the process of mechanization of diploidization, where polyploid will behave like a diploid. I think many of you know that. That's why I'm not going in detail. The PH gene is very important gene which suppresses the homologous pairing. Because of that, your wheat will behave like a diploid. That is only called as diploidization. And we'll see the depolarization is a very important mechanism in the wheat. Then, when you come to wide hybridization, we will discuss wide hybridization later, and we'll cover another type of polyploids so that I'll cover numerical chromosomal aberrations. Then we'll shift to wide hybridization. Okay. Then we discussed aneuploids, and we have discussed some case studies in wheat, how we can develop alien monosomic lines, disomic lines, and what is the use in the resistant breeding. We will discuss now what is euploids. What is euploid? In the euploid, we have haploid, we have triploid, we have tetraploid, pentaploid, we have so many. Okay, then what is haploid? We have discussed how to develop haploid, what is the use of haploid. That's why I'm not going in detail here. Okay, then we have triploid. We will discuss about triploids. What is the use of triploids? Why we need to develop a triploids? Okay. We'll discuss euploids. Okay, Adams classification. In euploids, we having autopolyploids and allopolyploids. Sorry, that is allopolyploids. Okay, we have auto, same genome is replicated many times, and we have different genome like allopolyploid. Where wheat is allopolyploid. See, writing the things is very difficult. Then we have first we will discuss about autopolyploids, so that it will be the auto triploid we will discuss. What is auto triploids? We having a 3x condition. The typical examples of auto triploids were your banana, spinach, and there are several examples like you can there are several examples like watermelon. What is this? This is the main source of your trisomics. Then the most of the triploids they're always sterile in nature, and only fertile triploid is spinach. That you should remember. Then they'll form trivalents. During the meiosis, they'll form trivalents and they form bivalents and they are also form univalents. And because of this, they actually, due to this formation of multivalents, what will happen? There is a mispairing of while chromosome the separation during the cell division will imbalance. That's why we get a partial sterility or completely sterility because of this. But in the case of diploidization, they'll act as a diploids. There is a proper segregation of chromosome. That's why wheats are fertile in nature. Then this is about triploid. We will discuss about tetraploids. What is tetraploids? We're having four copies of a single genome. Like it's a 4x. You take 4x. There are several examples of tetraploids. We having alpha alpha, and we have groundnut. Many times asked in the exam. Please remember the example. Groundnut, or it may be potato, or it may be coffee, grasses. There are several examples for there. Means they can form quadrivalents. Quadrivalents. They form univalent, trivalent, and they also form two bivalents. This is what cytological behavior of tetraploids in this one. Okay, and we have another typical type of autopolyploids. It is called as this autohexaploid. In autohexaploids, it is just single copy of a genome. It's repeated 6 times. Like we having is one example called as sweet potato. Then, what is the genetic consequence of this autopolyploidy? Genetic consequences of autopolyploidy. One is we having we know that when there is a, for example, you take alpha groundnut only. We having A, B, B, D, D. There's several copies were there. You think that all A copies. See, there will be increase in the proportion of the alleles. This having the dosage effect means the number of dose will be increases. Then definitely there will be increase in the their phenotype, gigantism. We will get a gigantism. Then higher rate of mutation. Because of this comes complexity, will get higher rate of mutations. And having high genetic variability in their population. High genetic variability in the population. This is what typical consequences of the autoploidy. Then here what will happen? Uh, Most of the times they are sterile in nature because of their formulation of multivalents. You'll see in autotetraploid condition like how double reduction happens in the next coming classes. We have in detail because I didn't prepare slides. Okay, I want to discuss about this one. Okay, this is about autohexaploids. And when it comes to allosomics, we have we have wheat, okay, allopolyploids, and there are different of allopolyploids. We will discuss in next coming classes. Okay, sorry.